Have you ever wondered how ecologists know that there are only a few dozen panthers left in Florida, or how wildlife biologists can count every fish in a giant lake? The answer isn't that they count every single one. Instead, they use a clever method called capture, mark, release, recapture to estimate the population size. This is a super important technique for understanding how populations change over time and for studying the human impacts on animal life. Let's explore this topic as though we're in your AP Environmental Science class. And to do that, we're going to simulate the capture, mark, release, and recapture right here in this lab with some beans and a little math. The core idea behind capture, mark, release, recapture is pretty simple. You grab a sample of a population, you mark them or add a tag to them, and then you put them back into the wild. Later, you grab another sample of that population and you count how many of your marked friends show up. By comparing the number of marked individuals to the total, you can use a simple equation to estimate the entire population size in that ecosystem. So now let's do our experiment. For our simulation, we aren't going to go out there and catch panthers. We are instead going to use two types of beans. We're using some black beans, about 500 of them, and yes, I counted every single one, and some white ones to be able to visually see our tagged friends from our technique we talked about earlier. We're also going to use a container like this clear beaker so that you can actually see what is happening during the simulation. So now let's do the simulation by counting about 500 black beans. I've already done this, so you don't have to sit here and watch me count all of them because it is a lot. So once that's done, you should probably figure out exactly the number that you counted and write that number down because we'll need it later for some. This will be important, like I said, later, because we're going to use that at the end to calculate how close our estimation is to the wild population. And that 500 beans you counted, that's going to be your wild population. It's like those panthers that are out there in the Everglades. And from our wild population, we're going to select 50 beans. These are going to count as our captured animals that we are going to swap out and tag. So now that we have our 50 beans, we are going to take those out and we're going to replace them as though they were chipped or microchipped with a small tag. And essentially, we are going to be replacing them with our white beans. So you take your tagged animals, you put them back into the population, and you mix it all up. So essentially, we are doing a catch and release with these beans. Imagine trying to count the number of fish in a lake. These first 50 beans represent 50 fish that were caught, tagged, and released back into the population. So now you can see we have our beans, we have some white beans mixed in with that original wild population. So you're gonna do this part without looking, and you wanna pick about 90 to 120 beans total. And you wanna do this kind of randomly so you get a nice mix of the population. Just make sure that you aren't letting any of them get too wild out of your sight. And as you see, I'm kind of grabbing handfuls, not really counting. We will count them eventually. So then this will be our second captured beans. Now let's actually count them. Let's start with counting our tagged friends. And so now from our second round of capturing, we have 14 that are recaptured. Now for the fun part, we got to count all of these animals or beans that have not been caught yet. And the number of beans that we had not caught before was 93. So the total beans that were captured this second round is 107 beans. Now that we have performed the second round of capturing, we have enough information to estimate the population size. Population size is estimated by seeing how many white beans have been recaptured. So now it's time for our favorite segment. We need to use the Lincoln-Peterson Index, which is an equation that scientists use to estimate the population size of animal species. And it looks something like this. We have P equals N1 times N2 divided by R. And within this equation, P is the estimated total size of our population. This number is what we are trying to find out. N1 is the number of individuals originally captured from our ecosystem. So that number will be the 50 white beans. And 2 is the total number captured in this trial. So our total captured was 107. And finally, R is the number of marked individuals recaptured in this trial. So if we're solving for P, we have all the necessary info, right? We can put in all our variables. And for R, we are putting in 14, which is our recaptured. After doing some math, we can determine that the estimated population size for this particular trial is about 382. And you can see that we originally had 500. And so you're gonna say, Dr. Simon, 382, that's a far off number from the 500 you had. Well, in all actuality, we're gonna have to do this same simulation 9 more times, and based on that data, we're gonna take the average. So bear with me and stay tuned while I do all of this counting. So now that we've done our 10 trials and figured out our estimated population for each trial, we can take the average of all of this to determine what the actual estimated population is of our original wild population. So when doing that average, I got about 505, which is really close to our original population that we counted. And this is why taking the average is so critical when doing these labs. Relying just on our original one trial wouldn't have been enough to actually get us to our accurate population. But why does this method work? Let's think critically about this. More recaptured marked beans in our sample means that the population is likely smaller because the marked beans make up a higher proportion of the sample, whereas fewer recaptured means that the population is likely larger since our marked beans are a smaller proportion of the total as a whole. Repeating trials allow us to gain a larger sample size and improve the accuracy by reducing random variations. And that's what we can see perfectly with our numbers. This simple simulation shows how ecologists use a smart, non-invasive method to gather critical data about ecosystems. Understanding how populations change helps scientists manage conservation efforts, protect endangered species, and maintain ecological balance. And that's it. Today you used some math and some hands-on experimentation to simulate how scientists estimate populations in nature. Lessons like these are what you'll encounter in your apes class, and there's plenty more in our course too. Use it as a companion to your course to prepare for quizzes and tests and get ahead of your peers for exam prep. Thank you for joining us today. If this video sparked your curiosity, please consider liking, sharing, and subscribing. Your support helps us bring more science to life. Until our next experiment, stay curious and keep exploring.